Well, good morning, good afternoon, and good evening, everyone, from a cold and wet Manchester here in England. But despite that, let me give you a very warm welcome to the third in our monthly series of webinars in the lead up to the World Cotton Research Conference next year. Our first speaker today is Professor Rattan Lal, and I'm sure that many of you know him. He is considered a living legend in the field of soil health, and we're truly blessed and honored to have him speak to us today. Professor Lal is the director of the Carbon Management and Sequestration Center at the Ohio State University and an adjunct professor of the University of Iceland. He has authored and co-authored 955 refereed journal articles and 543 book chapters, and has written 22 and edited and co-edited 76 books. He was included in the Thomson Reuters list of the world's most influential scientific minds, 2014 to 16, and he is amongst Clarivat's highly cited researchers in agriculture. He has received an honoris causa degree from 7 universities in Europe, USA, and Asia, the Medal of Honor from UIMP, Santander, Spain, the Distinguished Service Medal of IUSS, and is a fellow of the 5 professional societies. He is a laureate of GCHERA, World Agricultural Prize 2018, the Glinka World Soil Prize in 2018, the Japan Prize 2019, the USA Aswathi IFFCO Prize in 2019, and the World Food Prize 2020. And I can only say that I'm glad that list doesn't go on, Professor Lal, but that's just fantastic. So with that, thank you, Professor Lal, for agreeing to speak to us today, and over to you.
Thank you so much for the invitation and for your very kind words of introduction. It's a great honor indeed to talk about this important topic, soil health and its management. I would like to define something about what soil is to begin with. Soil is a living entity. It's a habitat for 25% of all terrestrial biodiversity. Furthermore, land misuse and soil mismanagement is a serious threat to soil health and biodiversity. Therefore, the objective of the management is to enhance and strengthen soil's capacity to support life, and soil organic matter content is the essence of soil health and its capacity to soil and its life support services that it provides for nature and humanity. So you can see the picture here of what a healthy, good soil should look like, full of organisms, good soil structure, and capacity to hold water and air and supply nutrients. So at this point, I like to define what soil health is. Soil's capacity as a dynamic and biologically active entity within natural managed landscape to sustain multiple ecosystem services, including net primary productivity, food and nutrition security, biodiversity, water purification and renewability, carbon sequestration, air quality and atmospheric chemistry, and elemental cycling, both for human and nature. Nature is an important part. Human are components of the nature. So it's both human and nature, not just human alone. Soil health is intricately interconnected with plant health that depends on it, animal health, human health, environmental health, and of course, planetary health. That link that the health of soil, plants, animal, human, environment, and planet is one and indivisible is important to recognize. Therefore, in addition to socioeconomic factors affecting food security, the food and nutrition security research must also address environmental and resource management issue, and the most important resource is soil. Going back to 1930s, It was Albrecht who said soil organic matter is one of our most important natural resources. Its unwise exploitation has been devastating, and it must be given its proper place in conservation policy as one of the major factors affecting crop production. Declining soil fertility due to lack of organic material, major nutrient elements, and trace minerals is responsible for poor crops and, in return, poor people. Health of our nation may be impossible to restore without first restoring health of our soil. And he was at that time president of the Soil Science Society of America. In fact, if you look at soil functionality and health, on that depends water resources, their quality and quantity, climate change mitigation, adaptation, and stabilization, food and nutrition security, both quantity and quality, and the biodiversity above and below ground. In fact, soil health is the engine of economic development anywhere globally. Cotton production, I don't need to give you any more statistics about it. You already know more than I do, but both worldwide and the US FAO stat shows that it's a very important economic crop globally. Therefore, management of soil health for productivity and sustainability of cotton is also very critical, like any other crop. In terms of the elemental cycling, biophysical and biogeochemical cycling, it's important to recognize the coupled cycling of water with that of carbon, with that of nitrogen, phosphorus, and sulfur, and other elements. They are all interconnected. Like John Muir said, in nature, when you try to pick up one thing, you find it hitched to everything else. And that's exactly what it is. And it is this interconnectivity, this coupled cycling, that is basis of many ecosystem services: carbon sequestration, water quality, biodiversity, net primary productivity. And it is the mismanagement of land and mismanagement of soil that can lead to decoupling. And it's decoupling that causes soil erosion, water pollution, emission of greenhouse gases, many other disservices. So the coupled cycling must be maintained. Soil organic carbon sequestration then means putting carbon back into the soil of a land unit through the plant, and the residue returned to the soil within that unit, so that the organic matter content can be increased to a range which is optimal. In general, when we convert natural ecosystem to agricultural ecosystem, the soil carbon stock decreases. It decreases because the input of biomass carbon is now less than it was in the natural ecosystem. Furthermore, soil erosion and mineralization risks are also higher. Therefore, in temperate climate, such as in Ohio, the soil organic matter content may decrease in an erosion-prone soil to about 50% over a 50-year period. If you are in a tropical climate, where cotton is most often grown, this decrease to 50% may happen in lesser period, such as 5 to 10 years. I worked in Nigeria for 20 years, and there the organic matter content would decrease to 50% within about 5 years. And more so if the soil erosion was a very serious problem, either by wind or water. Now, at this point, if the erosion is stopped, soil organic carbon content may reach a steady state level, a stable level. It may not change. But if we adopt better agricultural practices, which increase the input of biomass carbon through manure, green manure, returning crop residue, other biomass, carbon content will reach an equilibrium level with the adoption of better practice over about 20-25-year period following a sigmoid curve. At this point, if we adopt another better management practice, which further increases the input of biomass carbon, it may reach a level what we had started with in another 25 to 20-year period. So over a generational scale of 25 to 50 years, it is possible through better management practices to reach the carbon level where we were when we converted native system into agricultural system. In some cases, it is possible to go beyond that, but those are rare circumstances. These are the conditions where under natural condition, natural ecosystem, there are limitations in soil, such as water retention in a dry climate, lack of phosphorus perhaps, acidity, aluminum, iron toxicity. Under those conditions, it is possible to go beyond. Otherwise, you can reach about the maximum level, often not always the same level, but somewhere close to it. And we can define several factors from this. One is if you determine the rate of the slope of this line, delta y or delta x, that gives you the rate of change of carbon in soil, rate of carbon sequestration. You can also define soil carbon sink capacity, where you are when you started and where you can get there. This is the maximum amount that it can be stored, so that's the carbon sink capacity. You can also calculate the mean residence time. How long can carbon stay in the soil? If you know the total stock divided by the flux, annual rate of change, you get the mean residence time. The question is, how do you achieve this? And there is no one practice that's universally applicable. We have 300,000 types of known soils, and therefore to assume that one practice is universally applicable is probably being naive. On the other hand, some basic concepts such as conservation agriculture, regenerative agriculture, integrated nutrient and pest management, input of biochar, agroforestry, desertification control, afforestation, reforestation, improved pasture management, water harvesting, recycling, improved farming system complex are some of the good practices which can lead to carbon sequestration. However, each of these practices has its own carbon and water footprint. Therefore, when you calculate the gross rate of carbon sequestration, which is the slope of this line, you must subtract from it the cost of doing business, the cost of carbon input, water, other fertilizers, whatever managed through these management practices. And through this possibility, the idea is to create a positive soil carbon budget. Soil is like a bank account. You can increase your bank account if you add into it more than you withdraw. Similarly, in soil, carbon stock can be increased if you add carbon into it more than you lose. And you lose carbon by erosion, leaching, decomposition. At each site, you must know what the losses are. Therefore, you add into the soil more than the losses. You also, of course, decrease losses as well. So the goal is carbon sequestration, gains more than the losses. Unfortunately, in most circumstances, especially in resource-poor farmers in developing country, what they put into the soil is always less than what is taken away from soil by erosion, decomposition, leaching. Therefore, soil keep losing carbon stock. Health of the soil degrades over time. We want to create a positive soil carbon budget. In terms of the management practices also have a cost, as I mentioned. For example, plowing, moldboard plowing takes about 15 kilogram of carbon as a diesel consumption. Chiseling, another 8 kilogram. Heavy disking, little more than 8. Standard disking, subsoiling, of course, very energy intensive. Diesel consumption is much more. Cultivation, rotary hoeing, probably less. So if you eliminate some of these plowing, soil, tillage kind of operations, manage them properly, you can save carbon emission. Fertilizers are very carbon intensive, especially nitrogen. It can be about 2 kilogram per kilogram of active nitrogen in terms of the fertilizers. So saving nitrogen, reducing its losses, growing nitrogen by cover cropping legume is also important. Phosphorus is relatively less nitrogen carbon intensive, so is potassium, but all of them have cost. Herbicides, when you use herbicides, some of the herbicide can be 10, 12 kilogram of carbon, especially when the herbicide is used through the energy from fossil fuel. Insecticide the same way, very intensive. So saving fertilizers, saving of herbicide, savings of insecticide can save carbon emission involved in those inputs. Herbicides, different types, atrazine, dicamba, glyphosate, very carbon intensive, paraquat, ICI product, also very intensive. So anything that you can save in these inputs is obviously important. That brings me to the question of regenerative agriculture. That's the agriculture, cotton production system, for example, which will restore soil health through restoration, recarbonization of the soil carbon stock. It's inspired by eco-innovation, powered by non-carbon energy, driven by a circular economy and green infrastructure, supported by the recarbonization of the terrestrial biosphere, both soil and vegetation, at the bedrock of sustainable development. The question is, how do you manage carbon stock? You provide a continuous soil cover, never leave the ground ever bare, minimize soil disturbance, especially plowing, use integrated nutrient management, and adopt complex rotation, cotton grown in rotation with other crops. The idea is to restore soil carbon stock and produce more from less inputs. And the law of return, Sir Albert Howard, he stated in the 40s, his lecture, in fact, to the Indian Science Academy in 1920 when he was based in Indore. He said that harvesting without returning is a robbery of the soil and a banditry, a particularly mean form of banditry, because it involves robbing of future generations which are not here to defend themselves, because we must return soil health to the next generation better than what we inherited from the previous generation. We can also, by improving organic matter content, save nitrogen. Here's an example of data from Europe. Increasing soil carbon stock from 0.5% to 1% could reduce nitrogen fertilizer input by 50% in some cases. In US, each additional percent of organic matter in a depleted soil would save 31 kilogram of nitrogen per hectare. And at a cost of $1.50 per kilogram of nitrogen, that would save you almost $50 per hectare in nitrogen fertilizer. Therefore, what is soil-centric farming? The idea is to put more carbon emphasis than on NPK. So it's a CNPK rather than NPK. The idea is soil farming rather than cotton farming. Soil farming would improve cotton production because of the better soil health. It will also save in water. Several people have calculated, if you increase carbon stock in a depleted soil, how much saving of water. For example, additional 35 water stress-free days can happen if you increase organic matter content. There are many other studies which have shown if you increase organic carbon content by 1%, plant available water capacity, PAWC, could increase perhaps between 1.5 to 2%. Several thousand liters of additional water stored in soil. Many examples of saving in water are available in literature. I give here some examples. Compost can hold, one says, study 20 times its weight in water. Whether that happens depends on where the compost is applied or what the characteristics of compost are. But there are several examples which indicate that. And one of the reasons for better water holding capacity is what we study in soil analysis, PF curves, soil moisture characteristics curve. It's a force exerted by soil to hold water versus the amount of water, the relationship. So when a soil has organic matter content at a critical optimum level, the curve may look like this, compared with a soil, same soil, but the organic matter content is depleted. So if you calculate the wilting point of the soil and take that out of the field capacity, the difference what we call the plant available water capacity. This shown here in the red bracket, plant available water capacity is more in soil with organic matter than with less organic matter content. Therefore, if you are to alleviate a drought, mulch farming, conservation agriculture, which will save irrigation, drip sub-irrigation, solar irrigation system, increasing soil organic carbon content to improve plant available water capacity, and at the same time, nitrogen use efficiency, as I already mentioned about that. In fact, the cotton yield, if I may put it CY, can be given by a formula evapotranspiration time transpiration divided by evapotranspiration time 1 over transpiration time the harvest index. And in this equation, you notice nutrients are not there because in a drier environment where cotton may be more often grown, water is really the most critical factor. So it's the water management through better soil health that really determine the yield of cotton or any other crop. See why in that equation, it does not indicate fertilizer and other practices because these other practices of fertilizer, pesticide management, they influence yield through water. Water is life in this case. Therefore, this equation indicates how important management of soil health is so that it can retain more water. The common thread in this equation was water. Everything else determined that the critical level of organic matter content is about 3 to 4%. Depends on soil. In some soil, it may be 2.5%, in other probably 4%, but somewhere in that range. And most soils in the developing countries have less than 0.5% organic matter content in the root zone. And therefore, quite a lot can be done by improving the organic carbon and therefore soil health. If you have a crop yield in an unfertilized soil versus fertilized soil, increases in both cases with increase in carbon stock, but response is more in an unfertilized soil than in a fertilized. Fertilizer would have a masking effect. The progressive increase in yield with increase in carbon pool, of course, decreases with increase until it reaches the optimum level. Beyond that, with 3-4%, as I mentioned already, there is not much change. So what is soil eco-intensification or soil farming that I was talking about? The idea is to produce more from less. Less land, less water, less fertilizer, less pesticides, less energy, less carbon emission, saving extra land for nature. We are using too much land already. 1.5 billion hectares globally for crop production and almost 4 billion hectares for raising livestock. There is no reason that we cannot manage those lands better and return some land back to nature, even with increasing population. Hopefully population can be decreased. So the green revolution, rather than input-based, fertilizers, variety, irrigation, the green revolution of the 21st century should be soil health-based, soil resilience, ecosystem-based, eco-efficiency, environment-based, and of course it must be science-based, science and management-driven. And that is soil farming rather than crop or animal farming. Therefore, what is sustainable soil management? Going back to Albert Howard's concept, replace whatever you remove, respond wisely to whatever is changed, predict what may happen both from anthropogenic and natural perturbations. So think about a cotton plant that we can communicate with, perhaps through molecular-based signal, like some of the insect do pheromones under different stress, biotic or abiotic. Plants emit some signal that we can detect remote sensely and intervene in a targeted way so that the damage is not done. Root system should be deeper, more prolific, probably some possibility of mycorrhizal and nutrient use efficiency, especially nitrogen and other nutrients, is improved. Soil should be always covered by mulch, grow a complex crop rotation, including a cover crop in an off-season, and where possible, integrating livestock and trees as much as possible. The goal is to improve soil organic matter content and soil biodiversity so that soils are disease suppressive soil. Healthy soil with a lot of biodiversity suppress plant diseases. And that is a good option to manage cotton as well. And if any additional water and nutrients are required, they should be supplied in a way directly to the plant root through drip fertigation, for example, at a stage when required, so that most of the water and nutrients are absorbed by plant rather than leak into the environment. And that brings me to the subject of farming carbon. Just like cotton can be bought and sold, carbon increased in the soil can also have an economic value, both direct and indirect. So growing soil carbon, increasing the amount, as I showed you how we could do it, increasing, thinking it was like a balance, a bank account, add more than what you lose. So it is a farmer commodity, create another income stream for farmer. So farming carbon would create an income and therefore alleviate rural poverty. Rates of carbon sequestration under different systems in cropland, no-till, semi-arid, subhumid region, maybe half a ton under ideal condition of carbon per hectare per year. In a drier climate, maybe even less. But it is over a generational time scale can certainly improve soil health. And if we could sequester carbon in all soils of the world, the technical potential could be as much as 2.5 gigaton of carbon per year. Now remember, please, this will not offset the fossil fuel emission. Fossil fuel emission this year is slightly less, but last year it was 10 gigaton plus 1.5 gigaton from deforestation. So anthropogenic emission was 11.5. Soil ideally have a potential 2.5. So you're talking about 15%, maybe 20% the most. So finding non-carbon fuel sources is very critical. If we did all the soil and vegetation together between now and 2100, next 8-year period, we have potential of adding into the terrestrial biosphere more than 330 gigaton. That translates into atmospheric CO2 drawdown of more than 150 parts per million of CO2. So by finding non-carbon fuel sources, let's say between now and 2030, 40, 50, and using soil and the terrestrial biosphere as a sink, humanity has a better future in managing the environment, including climate change. There is a societal value of carbon. I was talking about farming carbon. The societal value, the ecosystem value, I'm not talking about the market value. The market value depends on demand and supply. This is the inherent, what it does for the nature. $130 per ton of carbon. That translates to $35 per ton of CO2. That translates into about 6-7 cents, little more than a nickel per pound of carbon. So if a farmer sequesters half a ton of carbon, that's worth $65 per hectare, $26 per acre. If farmers sequesters 1/3 of a ton, that's $43 per hectare, about $18 per acre. That brings me to the question of rights of soil, rights of nature. Soil is a living entity, just as the universal rights of human, rights of animals, rights of panda, rights of butterfly, rights of other living things, there must also be rights of soil. After all, it's a home for more than 25% of biodiversity, all biodiversity. Therefore, being the essence of all life, soil must have the rights to be protected, restored, thrive, and managed judiciously. And deliberate attempt to degrade, pollute, and destroy services of soil should have some legal connotations. Simply because you own it as a property doesn't mean that it can be destroyed. In that concept, agriculture has a very bright future. More changes will occur between 2020 and 2050 than have happened over the past 12,000 years since the onset of settled agriculture. And that will bring out a lot of things. Carbon in soil and trees, etc., will be a crop. Like a crop, it can also be traded through farming carbon. Therefore, rejuvenating soil health as a basis of soil-centric agriculture, that four-legged stool, conservation agriculture package for cotton and other crops, integrated nutrient pest management, drip sub-fertigation, crop residue management properly so it's not burnt, it's returned back. Even the urban waste can be composted and returned back to the land. And of course, cover cropping as a part of crop diversification. And all these things can be glued together through political governance, through political willpower, by translating science that we know into action, and by paying farmers not subsidies, but for ecosystem services that they create at a price that I just mentioned. And ecosystem services payment is not a handout, it's not a charity, it's not a donation. If farmers don't do it, they do not get it. Those who do it, they are rewarded properly by the society. I would like to mention that if I asked a question, what would I suggest to mitigate global warming, purify air and water, and the global hunger and malnutrition, the logical response would be to change the way soils are taken for granted and abused to produce, transport, process, and consume food, and misused to procure feed, fiber, fuel, bricks, and other ecosystem services. Therefore, a prudent strategy would imply making soil, water, and agriculture an integral part of the solution, and empowering farmers and land managers to produce more and more from less and less by reducing waste, increasing the eco-efficiency, restoring degraded soil, afforesting denuded land, and saving some soil and water land for nature, not taking more away from nature. And I think world peace depends on that. Imagine soil degrading by extractive farming practices, by erosion, by other pollution of industrial waste deposition. The soil carbon stock decreases, the nutrient stock decreases, soil structure declines, soil loses resilience, drought and other ecosystem declines, and function and services lead to loss of biodiversity, and soil does not produce what it's supposed to produce. That leads to climate refugees, leads to soil refugees. It causes hunger, malnutrition, political unrest, civil strife. Last year, we had 80 million refugees. I don't need to tell you what happened when the Mediterranean cannot hold the refugees back. This is a vicious cycle caused by soil degradation. And that cycle has to be broken through restorative policy. So if somebody asks me what is the most important part to bring world peace and stability, I would say degrading soils, denuding landscape, recurring drought, intensifying heat waves, low productivity, perpetual hunger, poor nutrition, increasing malnutrition, marginal living, desperateness are the biggest threat to global peace and security. These are the weapons of mass destruction. 25,000 people die per day, 17 per minute, because soil does not support them. And that need to be addressed. If I were to design a coin, that coin will have a logo in soil we trust. Thank you so much. I'll be glad to answer any question. Can you hear me?
Yeah, Kai, you are muted, Kai.
Thank you, Professor Lal, for that very interesting talk. I found it very, very interesting and absolutely fascinating. And I think you shown right at the end how this subject of soil health is arguably the most important subject, not just affecting cotton, but mankind in general. And thank you for putting that over. Now we have some questions, so I'm just going to start with one here. So we have one from Venu, who's from Nagpur in India. Will the organic carbon content of a nearby natural forest in the locality provide an idea of the upper limit to which soil organic matter of an agricultural system can be improved through good soil management interventions?
The answer is yes. In Ohio, we have 3 experimental sites where we started doing conservation agriculture, returning biomass back to the soil in 1960. And we have a wooded plot next door, and that is what we have been comparing with. And in 60 years since 1960, we are not quite there, but 90% we have reached that level. Rothamsted experiment also have wilderness plots left under native vegetation as a baseline. In most cases, yes. But if you're dealing with a desert climate, or you're dealing with a soil which has a low nutrient reserve, such as due to acidity, cerrado, some of the soils in the Amazon come under that category, then that is not the best option. But most cases, you are correct. Wooded area could be the baseline.
Thank you. The next question is from Akhtar Ruzaman from Bangladesh. As a deep-rooted crop, can cotton help carbon sequestration at the deeper part of the soil?
Absolutely. Anything which is deep-rooted, carbon is put by the roots into the subsoil layer. Subsoil are away from the zone of disturbance, away from erosion and other things. Yes, pigeon pea, I was growing in Africa, very deep root systems. Another cover crop, I was growing stylos, Townsville stylo from Brisbane, also very deep rooted. Anything which is deep rooted, trees, perennial, cotton, is a good thing. Yes.
Thank you. And also from Bangladesh, from Dr. Tabib, how does global warming impact on carbon emissions and soil fertility in hot and humid countries?
That is a very important question. Van't Hoff law says every 10 degrees centigrade increase in temperature doubles the rate of reaction. So rate of decomposition of organic matter will increase with increase in temperature. Therefore, remember that balance in which I had erosion, mineralization, decomposition. So both erosion hazard will increase with climate change and the decomposition rate will increase. Therefore, taking precaution now so that we do not go beyond the tipping point of 2 degrees Celsius, I think it's a very prudent strategy. We still can achieve 1.5 degree limit that was proposed at COP21 in Paris. We still can do it. But once it goes 2 degrees, 3 degrees centigrade, some places, then we are in serious trouble. Not only agricultural land, but also the permafrost soil, frozen soils. That can also thaw and cause problems. So let's hope that some action can be taken throughout the world by all countries and put carbon back in the terrestrial biosphere, soil, trees, wetland, vegetation. That's a good strategy. Thank you.
Thank you. The next one is from Rajiv Barua. His question is, how do we move to ecosystem services? In India today, the fertilizer subsidy is to the tune of 70,000 to 80,000 crores.
I have been watching news. Unfortunately, the farmers from Punjab and Haryana blocking traffic in and out of Delhi. I think the grievance is that they are not allowed to burn crop residue, and some of the subsidies for irrigation and fertilizer are being withdrawn. That is something, is a bitter pill have to be swallowed. But if we say to farmer, if you leave the crop residue and you do not burn it, you improve water quality and renewability of water, and you do not emit the gases through these disservices that are created, and we'll pay you for them, I think that's the answer. Substitute subsidies with payment for ecosystem services. If you do it, you get it. If you don't do it, you don't get it. And I think that is eventually the possibility. It needs to be done globally. In fact, Mr. Stefan Le Foll, who proposed the program 4 per mille in the Paris summit, I talked to him. He came to Ohio State before that announcement was made in Paris. My suggestion was, out of $100 billion they were requesting, 60, 70 billion should be devoted for farm ecosystem services. They didn't get 100 billion, they got 1 billion, and even that was withdrawn. But we have to be willing to pay. If we don't pay to farmers, it's not going to happen.
Thank you. And from Dr. Yenyamula, he asks, in the tropics, how difficult is it to build soil organic matter? Are there any effective strategies?
Organic matter build-up rate decreases with increase in temperatures. So the tropical soils do have a lower rate of increase than the temperate region soils have. There's no question about it. It's a bigger challenge. But tropical soils are also more degraded. So there's a more sink capacity. For example, soils of South Asia, loss of carbon from the original level, that was the very first question asked, has been 70, 80, 90% in some cases. So the sink capacity is also more there. Yes, it's a more challenge. Yes, you need to require more input, but there are many data from ICAR, from Central Asia, also from Sub-Saharan Africa, my own experiments. I did it in Africa for 20 years. Also many students working in Brazil. Yes, you can have carbon sequestration in soils of the tropic, provided that you add more than what you lose, and provided you decrease the losses by erosion, keep the soil covered and cool. That requires change of thinking, change of philosophy. We are used to keeping the plowed, bare, clean, bare ground surface. No, keep the ground covered, decrease the temperature of the soil, and mulching can decrease temperature in the summer in northwestern India by as much as 15, 20 degrees Celsius. Here is how to decrease the rate of decomposition. It can be done, it's challenging, and that challenge has to be accepted and mindset changed. Yes, we will do it.
Thank you. And from Dr. Ali Ahmed, can you please elaborate the importance of vermicompost for soil health?
Any compost is a great idea. Vermicompost means there are more earthworms. Remember, I have a picture which I showed you in the very beginning, what a good soil looked like, and earthworms are part of that. By the way, not many people know Darwin's first book was not on origin of species. His first book was on earthworms, origin of the molds. And he called earthworm the intestines of the earth. So vermicompost, which will also increase earthworm population, but please remember earthworms, like any other biotic organisms, require food. So taking away the crop residue, burning it, taking away the dung and burning it, not you putting it back on the soil, that's the banditry. Remember I was talking Albert Howard. And by the way, Albert Howard was based in Indore, India, and he developed the compost pit called Indore pit. And that was in 1920s, 1930s. It's a good idea to go back and see the design, and it's the same what you're calling the vermicompost now.
Thank you. And here's a question from Dr. Sabesh. I'm going to try and summarize this. Hybrid cotton in India is input intensive and burdensome on the soils. What is the way forward?
I think it's not as important, that's my personal belief, what is grown, but how it is grown. That's the key part. So regardless whether it's a hybrid or not hybrid, how you grow it If you grow it by conservation agriculture, which is a regenerative system which restores soil health, that is the part which is important. When soil organic carbon content is increased, I showed you how much nitrogen do you save, how much water you save, so how much pesticide you save because soil health has been improved. So having better soil health will reduce the dependence on the inputs in all crops, regardless of what that crop is. And once the soil health is degraded, then you try to restore that by substituting with chemicals, pesticides and herbicides and tillage. That's a symptomatic treatment of the problem. Go back to the soil where you grow cotton and find out if the organic carbon content is 0.1%, and I have monitored in some soil that number. You are going to dump chemicals, fertilizer, and soil is non-responsive. The idea is to make soil responsive to inputs, improve the efficiency.
Thank you. And this one's from Dr. Velmarorangi. Hopefully I've pronounced that correctly. What is the minimum threshold level for soil organic carbon with relation to crop yield?
I gave you a graph. You may remember I said that there is no A point on that graph. There's a range. And the range can be for tropical soil, perhaps between 2% organic matter to 3%. For temperate zone soil, it may be 3% to 4.5%, 5%. There is no one fixed point. And there's a range, and range also depends on texture of the soil, mineralogy of the soil. But by and large, if you're talking about Southern Asia, India, Nepal, Pakistan, Bangladesh, and so forth, you're talking about organic matter content somewhere between 2.5 to 3.5%. By the way, I keep on changing organic carbon and organic matter. Organic matter has 50% organic carbon. So when I say organic matter 3%, organic carbon is 1.5%. So that's the conversion factor. So that's a range, and that range maintaining will reduce the input dependence, fertilizer, pesticides, irrigation, water, and so forth. But please consult your local soil testing lab and make sure that you know your status of the soil at least to 30 centimeter depth, and try to improve. Overnight improvement is not going to happen. As I mentioned, takes 15, 20 years. But if you begin today, in 3, 4, 5 years, you'll see the difference.
Thank you. So we have one question in French, which I'll wait for that to be translated for me. So the next question is from Kate Hake from Cotton Incorporated. For growers of continuous cotton with full tillage, no rotation and no cover crops, what is the first most important step to take to improve their soils?
I would suggest begin with growing a cover crop so that you can improve the soil structural properties. A deep-rooted cover crop, aggressive cover crop, which produces a lot of biomass, especially the subsoil. And then after a cover crop, 1 or 2 seasons, I know farmers need income. That is where the payment for ecosystem services would come in. And then after 1 or 2 seasons, when soil structure has been improved, soil biotic activity improved, and while you are growing cover crop, hopefully adding some manure, compost, vermicompost, etc., then begin growing cotton by conservation agriculture system. If you grow cotton in a degraded, compacted soil, you are bound to fail. And that is when people come back and say, oh, we told you it doesn't work. The question is not it doesn't work, the question is how to make it work. Improve soil structure, soil health of degraded soil, and then you can get off that cycle of plowing and degrading and redegrading. We need to get out of that. So starting point should be improving soils. I want to give you an example for Africa. We were clearing forest, and immediately after clearing, tremendous serious erosion problem. So the idea was grow a cover crop. We were growing Mucuna utilis, kudzu, things of this nature. They produce 10, 20 tons of biomass. Leave the biomass on the ground, continuously cover, then grow crop through it. That's a system to adopt.
Okay, thank you. And from Dr. Sangram Shavan, is it true that cereals are more carbon consuming than other crops?
In general, that is true. Cereals require more cotton, does not require as much as wheat and rice and corn and sorghum. Cereals are what they call soil depleting crops compared to legumes and other perennials. But as I mentioned before, it is not as important what you grow, but how you grow it. So a corn which requires, which is considered erosion promoting and soil degrading crop, if grown by conservation agriculture with a mulch cover crop in the rotation cycle, no tillage, it can also be as good as a soil conserving crop such as a legume. So what you grow is less important than how you grow it. But in general, what you said is correct.
And from Mohammed Jahangir Alam, how can we improve soil health in drought areas where the soil is acidic?
First, overcome the acidity problem. If the soil is acidic, then somehow acidity neutralization is very critical. So liming, compost, manure, um, everything goes back to the carbon stock. If soil is in a dry climate and is also acidic, overcome the water limitation and overcome the acidity. Neutralize acidity by some kind of polyvalent cation, calcium, magnesium. Those are very critical. Probably acidity may be in aluminum, iron, manganese, hydrogen. And if the soils are also at the same time drought prone, you got to overcome those two problems as well. But once the soil has a better organic carbon content, soil biotic activity, please remember soil organism, whether it's earthworm, termites, centipedes, millipedes, microorganisms, they are very critical to improving soil health, and they require food, and their food, like our food, is biomass material. So biomass is the basis.
Thank you. So we're if Caroline's on, perhaps she can translate the French question. Caroline, can you do that? I'm not sure she can hear me. Okay, so there's one from Dr. Sain. What is the role of AM in soil carbon?
What is AN? Could you tell me that, please?
I don't know.
Are you talking about ammonical nitrogen?
It's AM. He's put AM in the
AM.
AM.
Somewhere somebody had to tell me what AM stands for.
Perhaps you can put it in the chat very quickly.
Yeah.
And let me take another question then.
Okay.
Right, it's I've got the answer, and it's arbuscular
Mycorrhiza, yes.
mycorrhizae. Yeah.
I had in my picture, if you noticed the picture, I had a plant in which I showed, and it underneath it said mycorrhiza, and that's, yes, they are very important. Any root inoculation of mycorrhiza would lead to, the word should not have been AM, it should have been AMF, mycorrhizae fungi, and arbuscular mycorrhizal fungi, that's AMF. And yes, if soils are devoid of mycorrhizal, then inoculation certainly helps.
Okay, let's take another one, which is, I'm trying to summarize this. Can huge incorporation of organic matter in the soil lead to CO2 emissions as soil respiration?
Please remember, soil is a living thing. And when I go to see my doctor every year, they put me on a treadmill, and they're trying to evaluate my health, and they put a mouthpiece so they can see how much CO2 I'm respiring. And more the CO2 I respire when I'm on a treadmill, the interpretation is better the health. Soil is the same way. A healthy soil has to respire. I killed a soil. I deliberately killed it, and there was no respiration, there was no loss, there was no CO2, there were no organisms, so all were dead. And dead soil does not produce any. So when people say that we are trying to stop decomposition of organic matter in soil so it does not respire, you are killing a soil. Over and above the respiration, we have to add organic matter more in the soil so that the balance is left behind soil. That is what I was showing you by balance, creating a positive carbon budget. I use the word mineralization, you are using the word respiration, they are the same thing. I have to provide, if a person wants to gain weight, they must eat more than what their metabolic respiration is. That's the only way they will gain weight. Soil is the same thing. Soil is a living thing. You must feed soil. When I said farm soil, you are feeding soil. You have to take care of the animals in soil. Do not starve them. They must respire like we respire. You don't want to suffocate them. You don't want to starve them. You don't want to kill them. Soil respiration is very important. A soil which is anaerobic, instead of doing CO2, it will respire methane. You don't want to do that either. So a healthy soil means 25% of the pore containing water, 25% air, about 3 to 4% organic matter that is continuously recycling. That's what a healthy soil is. Let it respire.
Okay, thank you. From Dr. Kumar, how important is potassium in cotton production, and how quickly does it deplete in soil if not replenished?
Potassium, in fact, one of the problems that has happened in India and other places in developing, nitrogen was subsidized. Therefore, nitrogen is much cheaper than phosphorus and potassium. So the problem is imbalance. You know, the Lebesgue's law of minima, the strength of the chain is determined by the weakest link. So you add nitrogen, but if there is no potassium, what can nitrogen do? The nitrogen use efficiency, unfortunately, is hardly 25, 30% in a best case scenario, because the other nutrients are limited. Yes, if potassium is not substituted, it will be called limiting. It means even nitrogen is not effectively utilized. So balanced nutrient application is the key thing, balanced. So when you do not have balanced NPK ratio, it's not a good management of soil production. In plant health, micronutrients are very important also. Zinc, iron, molybdenum, these are, and sometimes we ignore zinc, iron, and these micronutrients. We have to also consider not only P and K, but also micro. Potassium is also good for drought tolerance. So soil test and find out whether the nutrient are in balanced supply. And that's very critical.
Okay, and this is the translation of the question in French. In which proportion the use of organic fertilizer, in which proportion can the use of organic fertilizer reduce the use of chemical fertilizer? So for example, for 5 tons of organic fertilizer on 1 hectare of cotton, what is the additional quantity of NPK needed?
It depends on the soil test. If your soil test indicates that there are already some nutrients and what you need for cotton, let's say you need for cotton 50 kilogram of nitrogen, and your farmyard manure supplied 40 kilogram, and soil had nothing, then you must add 10 kilogram from your fertilizers. And that's exactly what the soil test is for. So soil test what is available in soil, what is crop requirement to produce the desired yield of cotton, and then whatever is available in manure, that is what the supplement should come from the fertilizer. Simply dumping fertilizer without knowing soil test is not a very good scientific method.
Thank you. And I'm very conscious that we're right up against time for you, Professor Lal. Keshav, if you feel that there's one other question amongst the many others that I haven't asked that you feel that should be asked, please jump in now. Otherwise, from me, Professor Lal, thank you very much indeed. It was a really, really fascinating lecture, and I'm deeply grateful to you once again.
Dr. Hughes, Dr. Kranti, I do want to thank you guys giving me the opportunity to talk to you. I hope we can answer the question of the audience some other time, but I appreciate this opportunity, and I wish you all a very happy Soil Day coming up on 5th of December.
Yes.
And I believe this was in honor of the World Soil Day. And by the way, the World Soil Day is in honor of the late King of Thailand, and his name was Bhumipal Atulyatej. My name, Sanskrit based, and you know the word Bhumi. Bhumi means soil. Bhumipal, like Gopal. Atulyatej, unique power of soil protector. That was King of Thailand. And the World Soil Day is celebrated on his birthday, 5th of December. So I wish you all a very happy Soil Day. Thank you again. I greatly appreciated the opportunity to talk to you.
Professor Lal, thank you so much. On behalf of the International Cotton Research, on behalf of the Cotton Researchers Association, and on behalf of the International Cotton Advisory Committee, and on behalf of all the participants, thank you so much. It was a brilliant day today and a brilliant talk. Thank you.
Yes. Thank you very much. And now we move on to our next speaker, who is Professor Bruce Tabashnik, who's going to talk on the topic of battling bollworm resistance to BT cotton. Now, Professor Tabashnik is one of the most influential scientists of our times in entomology and biological sciences. He's led the Department of Entomology at the University of Arizona for 24 years. Professor Tabashnik's research team studies the evolution and management of insect resistance to insecticides and transgenic plants. The Web of Science Clarivet Analytics recognized his work as highly cited in 2018 and 19, and Professor Tabashnik is a fellow of the Royal Society of Entomology here in the UK. He's also the recipient of several awards, which include the Nanyao Su Award for Innovation and Creativity in Entomology from the Entomological Society of America, the Koffler Prize in Research, Scholarship, Creative Activity from the University of Arizona, and he recently won the Plant Insect Ecosystems Lifetime Achievement Award in Entomology from the Entomological Society of America. Bruce, over to you.
Thank you so much, Kai, for that wonderful introduction. It's going to be a tough act to follow. That was an incredible presentation by Professor Lal, but I'm going to do my best. I'm going to switch to sharing my screen here. Let's see. I'm delighted and honored to have the opportunity to talk with you today about battling bollworm resistance to BT cotton, a tale of 3 countries. And with COVID, our ability to travel has been greatly restricted. But today I want to take you along with me on a journey to 3 countries, the United States, India, and China, to tell this story. I want to start by thanking my many wonderful collaborators and also funding sources. Let's turn back the clock for a minute to 1984. At that time, the number of insect species with 1 or more populations resistant to an insecticide had climbed to 462. At the same time, the average cost of developing a new pesticide had soared to $45 million. So things were looking pretty bleak with these conventional insecticides. So science turned instead to a natural way of controlling insects, a bacterium called Bacillus thuringiensis, or BT for short. And like I used to tell my mom, yes, Mom, BT, those are my initials, but no, I didn't invent this, I just study it. BT is a natural widespread bacterial pathogen of some insects, and BT proteins have been used in sprays for more than 80 years, nearly a century, to control insects that vector disease and also are pests in forests and crops. The great thing about BT for pest control is that the BT proteins are not harmful to most non-target organisms, including natural enemies of the pests, and most importantly, people. And about 30 years ago, the genes encoding BT proteins were first engineered into crops, allowing them to produce their own BT proteins for insect protection. And you can see it in the image at the top, the crystal inside the cell, the bacterial cell, that's the BT protein. And then on the bottom, a bunch of purified crystalline BT proteins, and they're called cry proteins for short. Oops. These BT crops have many benefits. They're highly effective against certain key target insect pests, and they can kill boring pests. Now, by boring pests, I don't mean dull and annoying, although they may well be that. I mean pests that bore inside plant parts and can't be reached by typical insecticide sprays. As I mentioned, BT crops that produce BT proteins have little or no toxicity to most other species. So these can help to reduce insecticide use, providing economic, environmental, and health benefits. With these wonderful benefits, the global adoption of BT crops has been one of the most rapid of any technology in the history of agriculture. Starting in 1996, with about 1 million hectares of BT crops grown worldwide and increasing to over 100 million hectares by 2018. And this has included 82% of the corn planted in the United States in the current year, and 94% of the cotton planted in India in 2019, about 12 million hectares of BT cotton planted there. But with this widespread adoption in the history of insects' ability to adapt to whatever we use to control them, about the time BT crops were first made commercially available, experts in this field predicted from modeling and the history of insecticide resistance that under worst case scenarios, we could see pest resistance to BT crops evolving in as few as 3 years. So the question that I've been addressing with my team for the last few decades is, can we apply evolutionary principles and knowledge about resistance to make BT crops more sustainable? And I hope that by the time I finish my talk this morning, you'll agree that the answer to this question is yes. So for the rest of the talk, I'm going to give you a definition of what I mean by resistance and quickly review the global data on resistance to BT crops. Then I'm going to summarize for you the theory underlying the refuge strategy for delaying evolution of resistance. And for the remainder, I'll focus on just one pest species, the pink bollworm, and how the outcomes with this pest have been remarkably different in the US, China, and India, the world's 3 leading cotton-producing countries, which collectively typically produce more than 60% of the world's cotton. So field-evolved or field-selected resistance is a genetically based decrease in susceptibility of an insect population to a toxin that's caused by exposure to a toxin in the field. And we can break field-evolved resistance to Bt crops into categories. The one I'm going to emphasize today is called practical resistance. And this is resistance that's severe enough to have practical consequences for pest management in the field. How do we know if populations are resistant? The main method involves lab bioassays. So first, insects are collected from the field. Their progeny are reared in the lab under controlled conditions. And the larvae, the immature stages, are tested on food that's been treated with a toxin or on the Bt plant material itself. And typically, mortality is recorded. So based on these kind of bioassays, monitoring that's been going on worldwide since Bt crops were first commercialized has shown that as of 2018, there were 22 cases of field-evolved resistance to Bt crops involving 9 major pests. 7 of these are lepidopterans, that is caterpillar pests, 2 are beetles. And collectively, these pests have evolved resistance to all of the 9 major cry toxins used in Bt crops worldwide. And these cases have been recorded from 6 countries. And you can see on the graph on the right that resistance didn't evolve in just 3 years as was predicted under the worst case scenario. The first case took about 6 years, was recorded in 2002. Now I'm going to briefly review the theory underlying the refuge strategy. We're going to consider the simplest possible case, that is, plants that produce a single Bt toxin and insect resistance that's conferred primarily or entirely by a single gene with 2 alleles, the R allele conferring resistance and the S allele susceptibility to the Bt protein. And although this is purposely simplified, it's reasonably realistic. So this gives us 3 genotypes, the SS susceptible homozygotes, the RS heterozygotes, and the RR resistant homozygotes. And here's what the refuge strategy looks like under ideal conditions. The first critical ideal condition is that the resistance of the insect is inherited as a recessive trait. Second, those R alleles, the alleles conferring resistance, are rare. Now it's a recessive trait, so only the extremely rare RR homozygotes are going to survive when they feed on the Bt crop. In this case, this pink rectangle representing the Bt cotton. But who are those rare RR homozygotes going to mate with? If we have a nearby refuge of non-Bt cotton, most of the individuals emerging from that refuge are going to be susceptibles. And those rare resistant homozygotes are going to mate mostly with those susceptibles. And here you see depicted a ratio of 6 susceptibles for every resistant homozygote, but more realistically, think of a ratio of 1,000 to 1 or even 1,000,000 to 1. So these matings are going to produce RS heterozygotes, but their inheritance of resistance is recessive. So those heterozygotes are going to be killed by Bt cotton. And many models predict, and a fair bit of evidence confirms, that when these conditions are met, a refuge of non-Bt cotton or another non-Bt crop can greatly delay the evolution of pest resistance. From here on out, I'm going to focus on pink bollworm, or sometimes I use PBW for short. The name of this species is Pectinophora gossypiella. You may know there are many bollworms that attack cotton. I'm focusing on just this one species for the rest of the presentation. The pink bollworm is a global pest of cotton. And when we started this work on resistance to Bt cotton in Arizona a few decades ago, we thought that the risk of this pest evolving resistance to Bt cotton in Arizona was high. Why? Because in the lab, we were able to select for resistance to Bt cotton in just 2 generations. And in the field, this pest feeds almost exclusively on cotton in Arizona. And since 1997, more than half of the cotton grown in Arizona has been Bt cotton. And this pest has 5 generations per year. I want to mention that the moth shown in the upper right corner, the female lays its eggs on the cotton bolls. The larvae hatch and feed within the cotton bolls. And if the infestation is sufficiently high, this pest can completely destroy the cotton yield. I want to share with you that our team's work has shown that the genetics of pink bollworm resistance to Bt toxins is similar in India, the US, and China. And in particular, this pest has recessive resistance to Cry1AC, the first Bt toxin used in Bt cotton, and to Cry2AB, the second Bt toxin used in Bt crops. And that's very important because that, as you've seen from the earlier slide, gives us a better chance for the refuge strategy to delay resistance. I'm going to give you my conclusions now in case I run out of time or you have something more important to do later. First of all, resistance management matters. We will see that refuges of non-Bt host plants can delay evolution of resistance to Bt crops. We're going to be looking at the same pest, the same crop, but very different management and remarkably different outcomes in the US, India, and China. I want to present a very important caveat, though, to keep in mind, a limitation here. This is a natural experiment. It's not controlled. So there certainly are other factors that differ among these 3 countries that could have contributed to the different outcomes. So here are the here's a summary of the 3 very different outcomes of pink bollworm resistance to Bt cotton in the world's top 3 cotton producing countries. In India, resistance evolved after 6 years to Cry1AC, and pink bollworm was resistant to both Cry1AC and Cry2AB in less than 10 years. In the US, completely opposite outcome. No resistance observed at all after 24 years. In fact, this pest is now eradicated. In China, an intriguing intermediate outcome. Resistance first rose a bit, then it dropped after planting of F2 cotton hybrids between Bt and non-Bt cotton increased the percentage of refuge non-Bt cotton plants to 25%. Now we're going to look at each of these 3 in a little more detail. We'll start with India. As I mentioned, yes, after less than 10 years, pink bollworm evolved resistance to both Cry1AC and Cry2AB. And we think a major factor contributing to this was that even though refuges were mandated, they were not widely grown. So there was very little non-Bt cotton. And I want to share with you this video. This is a harvesting machine. This is from India, Gujarat in 2015. It's supposed to be harvesting cotton, but you can see the machine is also filled with pink bollworm. Not the outcome that anyone wants. And here are some data generously shared with me by Dr. Kranthi, showing pink bollworm resistance to what's called Bollgard-2 cotton. It produces 2 toxins, Cry1AC and Cry2AB. And if you look on the y-axis, you'll see the percentage of bolls of this type of cotton infested by pink bollworm larvae. In 2010 and 2011, infestation across the 22 sites studied was very low. Only 1 case where more than 20% was seen. Most of the infestation was 0. Dramatic change, 2015 and 2016, high levels of infestation across nearly all the sites, and even 100% infestation of this Bollgard 2 cotton at some sites. Now I want to contrast this with the story in Arizona. No pink bollworm resistance to BT cotton has occurred after 24 years. In the first decade, from 1996 to 2005, farmers planted non-BT cotton refuge sufficiently to delay evolution of resistance in this pest. And then starting in 2006, the farmers switched to a novel strategy. A multi-tactic eradication program was initiated that included sterile releases of sterile moths. And I'm going to tell you about that in more detail in just a moment. And this program was successful, leading to declaration of eradication of the pink bollworm from the continental United States in 2018. Well, here's how this eradication tactics worked. The key here is eliminating, for the most part, the non-BT cotton refuge and replacing it instead with billions of sterile moths released from airplanes over every cotton field of the state of Arizona several times a week. So now when those rare resistant homozygotes emerge from the BT cotton, who will they mate with? They're going to mate with primarily the sterile moths released from the airplanes. But these matings with sterile moths produce no fertile progeny. So we did some computer simulations looking at how the population dynamics of this pest would be affected in principle by BT cotton alone, sterile moth releases, or the combination of the two. In this figure, the wild moths, the pink bollworm moths, in millions are represented on the y-axis on a log scale. Let's look first at the scenario with only steriles released. In this case, the steriles are not sufficient to suppress the population. Population size increases until it reaches its carrying capacity. What if we use BT cotton alone? And the refuge percentage here is the actual percentage observed in the field, which was 7% non-BT cotton, 93% BT cotton. Here the model predicts that we'll see a gradual decrease in the population size. However, this decrease is so slow that it increases the probability that the pest will evolve resistance before eradication occurs. On the other hand, if we combine both tactics, as you see here with the black line and the black circles, eradication is predicted to occur in a little over 3 years. In other words, this combination of control tactics is synergistic. So here's what actually happened. There were 2 to 3 releases of the sterile moths per week per field. I want to mention that these sterile moths were produced in essentially a pink bollworm factory that was in the Phoenix area of Arizona. More than 11 billion, that's billion with a B, moths were released from 2006 to 2014. And we tracked pink bollworm susceptibility to Cry1AC and BT cotton from 1997 to 2018. Susceptibility did not decrease in Arizona throughout these 2 decades. We used 3 different methods to track susceptibility. All 3 showed no decrease in susceptibility. The first is field efficacy, which compares the density of pink bollworm larvae in paired BT and non-BT cotton fields. We used standard lab bioassays with Cry1AC in the diet. And we also screened DNA for cadherin resistance alleles that are tightly linked with resistance to Cry1AC and BT cotton in this pest. So here's what happened to the density of the pest. This is the pink bollworm larvae in non-BT cotton bolls. That's shown on the y-axis on a log scale, the percentage of infested non-BT cotton bolls. You can see before the eradication, there was perhaps a slight decline from 1997 to 2005. When the eradication program, the density of pink bollworm larvae in non-BT cotton bolls plummeted. The last time any pink bollworm larvae were detected in non-BT cotton bolls was in 2009. From 2010 to 2018, our screening of over 86,000 bolls detected no pink bollworm larvae. So the impact of BT cotton in the southwestern US and northern Mexico has been tremendously positive. I focused on Arizona in my talk because that's where I am and that's where we collected the most intense and complete data set. But this program actually extended to several other states in the southwestern United States and also northern Mexico where cotton is grown. So the pink bollworm eradication was declared in 2018. Insecticide use against this pest has been completely eliminated, and this has provided economic, environmental, and health benefits. Now we're going to shift gears and travel halfway across the world to the Yangtze River Valley of China, where millions of small-scale farmers grow cotton. And BT cotton producing Cry1AC was introduced there in 2000, and refuges of non-BT cotton fields are not required there. And since 2008, fields of non-BT cotton have accounted for less than 10% of cotton. So under these conditions, we would expect resistance to evolve in the pink bollworm. Oops, having a little trouble advancing here. Okay, let me try it a different way. So we started tracking pink bollworm resistance to Cry1AC in China in 2005, and you can see in the graphs on the left, the one at the top, the populations tested that had one or more survivors. The percentage of the populations tested was 0, 2005, 2006, and 2007. This was a quite extensive study involving 17 sites in 6 provinces and testing more than 66,000 larvae. Now, the percentage of populations with one or more resistant individuals jumped in 2008 to about 40% and then to over 60% in 2008. The percentage of resistant individuals was 0 in 2005, 2006, and 2007, as you can see in the lower graph, but then it climbed to about 2.5%. So the average population had about 2.5% resistant individuals in 2009. And given the high percentage of BT cotton, we expected resistance to increase quickly thereafter. But we were wrong. The resistance went away. Susceptibility was restored. You can see that by 2011, none of the populations had any survivors at our test concentration. And the percentage of resistant individuals overall also dropped to 0 by 2011 and remained at 0 until 2015. So we asked ourselves, well, why are we wrong? What the heck is going on here? My collaborators in China knew the inside story, something that wasn't well known publicly or widely, and that was that farmers were planting F2 cotton hybrids from BT crossed with non-BT cotton. Why? Well, one of the reasons is that the F1 and F2 hybrids have a higher yield than the parent varieties. Not only that, cotton as you know, self-pollinates. So the F1 had to be hand pollinated, which is expensive. And this yields 100% BT cotton. However, once you have the F1, it's much less expensive to produce F2 and subsequent hybrids just by allowing the cotton to self. But these F2 and subsequent generations are going to have only 25% non-BT cotton plants and 75% BT cotton. So when the seeds from these F2 hybrids are planted, it provides a built-in refuge within cotton fields of 25% non-BT cotton plants. So we hypothesized first that planting of the F2 hybrids increased in 2010 and after. And second, that this increased planting of non-BT cotton refuge plants provided by the F2 hybrids is sufficient to explain the observed reversal of resistance. So to test the first hypothesis, our team measured the Cry1AC content in 16 pure varieties and 68 hybrids, the 84 types of cotton that were the most popular in the Yangtze River Valley during the period we were studying. Overall, 14,000 seeds were tested individually. And the BT cotton, 6 varieties, 99% tested positive for Cry1AC. A little bit surprising, the non-BT cotton, 10 varieties tested had 23% on average positive for Cry1AC, which indicates contamination of the non-BT cotton seed supply with BT cotton seeds. The F1 hybrids had 96% positive for Cry1AC, which is about as expected. The F2 hybrids had 67% positive for Cry1AC, which did not differ significantly from the 75% expected. Knowing which types of cotton were planted and the percentage of Cry1AC in each of the types allowed us to calculate the percentage of non-BT cotton planted in any given year, which is shown here on the y-axis. So in 2004, you can see the percentage of non-BT cotton was high, close to 40%. That's the black circles and the black lines. But this decreased to 2009, and then it increased dramatically in 2010 and increased slightly thereafter. Well, why did this occur? This occurred because of a dramatic increase in the planting of these F2 hybrids, which is shown by the purple squares. This increased from close to 0% in 2004, a little bit to 2009, but then it jumped between 2009 and 2010 to close to 20%, and then approached 20% thereafter. So this jump in the percentage of F2 hybrids corresponds with the reversal of resistance. We use simulation modeling to see if the observed pattern could be explained by the F2 hybrid plants within the fields. And then we have 2 predictions. We have on the y-axis the percentage of resistant individuals going from 0 to 100%. The red dashed line is the prediction if only the non-BT plants in fields of non-BT cotton act as refuges. The blue dashed line with the blue boxes is what's predicted if non-BT plants in F2 hybrid fields act as refuges. You can see the predictions are opposite. If only the non-BT cotton plants and non-BT cotton fields act as refuges, we expect resistance to evolve quickly. But if those F2 hybrid plants if those non-BT plants in F2 hybrid field act as refuges, then we expect resistance to be delayed. I'm going to superimpose the observed data, what actually happened, these black triangles, and you can see it corresponds well with the prediction if non-BT plants and F2 hybrid fields do act as refuges. So these results imply that the non-BT plants and F2 hybrid fields were sufficient to explain the observed pattern. So one of the positive outcomes of this, as you can see on the top, the pink bollworm population density decreased 96%. We have on the y-axis eggs per 100 cotton plants, that's pink bollworm eggs, declining 96% from 1995 to 2015. And the insecticide sprays targeting pink bollworm decreased by 69%. So the impact of BT cotton in China has been very positive. I want to point out that these F2 hybrids were planted voluntarily, not to manage resistance. We think growers planted these because of the lower seed cost and the higher yield. The net outcome has been the pink bollworm's been suppressed. Again, insecticide use against this pest greatly reduced with economic, environmental, and health benefits. Well, what about the future? I think we're going to see more transgenic crops for insect control, new BT toxins. The VIP is a different kind of BT toxin that's already been incorporated in some widely grown crops, including cotton. There are other new BT toxins in the pipeline. I think we're going to see new pests targeted by BT crops, not just coleopteran and lepidopteran pests, but also others like lygus bugs and thrips. I think we're going to see BT expand into other kinds of crops, not just cotton and corn, which have been the mainstays. And already BT soybean is grown widely in Brazil, and we have now BT eggplant being grown in Bangladesh. We're going to see BT not just by itself, but with RNA interference. This type of plant with corn is already in the pipeline in the United States. And I think we're also going to see insecticidal proteins incorporated in transgenic plants that are derived from bacteria other than BT. So in conclusion, we've seen the first cases of resistance to BT crops, but by no means the last. Thank you very much. I welcome any questions.
Well, thank you, Bruce. We had a bit of a problem trying to unmute ourselves.
Yeah.
Thank you very much indeed. I'm going to hand this over to Dr. Kranthi, who is much more able than me to ask all these questions. Keshav.
Thank you, Kai. There's this question from Dr. Hanchinal from India. He says, the pink bollworm developed resistance to BT and farmers are struggling to manage the bollworm and again have gone back to the non-BT era. They're also using a lot of chemical sprays. Is it feasible to have the sterile technique program for pink bollworm in India, or is there any other way how we can control this pest?
I would love to see the steriles used in India. It would not be easy because the vast area of cotton grown in India, 12 million hectares of cotton, of BT cotton alone, which is 94% of the total, but anyway, more than 12 million hectares. That's a whole lot more than Arizona. So logistically, it would not be easy. It might be possible in some limited locations. Farmers in India have returned to integrated pest management practices that they had used before BT cotton. So, and at the moment, I think Dr. Kranthi can probably tell you better than I can, but I think that is helping. And this involves things such as not leaving the crop in the field as long, and other cultural practices that reduce pink bollworm. The insecticide sprays are not particularly effective, but they can be used judiciously in combination with other controls. And I'm hoping that we'll develop BT cotton with some new toxins that would be effective against pink bollworm that are resistant to the BT toxins that have been used before. We've tested some in my lab that kill the ones that are resistant in the lab, and I'd love to find out if they would also kill the resistant insects from India. I think they would, because the genetic basis of resistance is similar in the field selected populations in India and the lab selected populations we've tested in Arizona.
Thank you. This question is from Dr. Dilip Monga, and he's asking a question on refugium bag. Incidentally, the government of India has introduced refugium bag strategy very recently. So his question is, can the pink bollworm resistance development in central and south zone of India be reversed over time with the use of refugium bag?
The short answer is no. In the short or even intermediate term, let's say 5 to 10 years, refuge in a bag is probably not going to help much. I'll tell you why. Refuges, as you saw when I outlined the ideal conditions, work best when resistance is rare. When resistance is rare, refuges can be very effective. That's because the susceptibles coming from the refuge can greatly outnumber the resistant insects that can survive on the BT crop. But by the time the resistance is common, so common that it's causing control problems, it's too late for the refuge strategy. So until we have new toxins for which resistance is rare, refuge in a bag is not going to help with pink bollworm resistance very much.
Thank you. Dr. Katagiri from India is asking two questions. His first question is, do the pink bollworm larvae feed on boll rind before entering the bolls? And the second question is, the hybrid cotton plants in India, they carry F2 seeds, which contain non-BT seeds. When pink bollworm larvae enter the bolls and when they feed on the non-BT seed, they survive and grow. How do these larvae impact resistance?
So let's see, was the first question whether the larvae feed on the boll as they enter?
Yes, do they feed on the boll wall, on the boll rind as they enter?
Yes, they do. Yes, they do. They have to. To get in, they have to chew their way into the boll, so they do feed on the boll wall. And the second question about the F2 seeds not containing BT, that in principle should have helped to delay resistance. But apparently it didn't help enough. That's the short answer.
Thank you. This question is from Dr. Santosh from India again. He's asking if, based on current cotton cultivation scenario in India, how many years could it take for Helicoverpa armigera to gain threshold resistance to Cryovenaceae and also to Cryovenaceae plus Cry2AB?
That's a great question. I might defer that question to you, Dr. Krinthi, because I think you know better than I do. What do you think?
Thank you. You're putting me in a spot, Bruce. But then, yeah, I think there is still a greater risk because there have been instances and reports of bollworms occurring on Bolgar 2, but these have been sporadic. Fortunately, because of a large number of other crops being grown, and also because Bolgar 2 was introduced timely in India, there's been a delay. But yes, you are the expert today, and I'd love to know what you tell us.
Well, I want to amplify something that you mentioned. So pink bollworm is different from Helicoverpa armigera, also called the cotton bollworm, because in India and most other places, Helicoverpa armigera has many other host plants that it feeds on. And those host plants can act as a refuge because they're non-BT plants for the most part, or sometimes entirely. So they can provide a very large refuge. So a large proportion of the food that Helicoverpa armigera are eating can be non-BT plants. So that's a great thing. And that's probably why we haven't seen Helicoverpa armigera resistance as severely in India as we see pink bollworm resistance, because pink bollworm relies almost exclusively on cotton as its host plant. But Dr. Kranthi's work, and I think some other monitoring, is showing what we call early signs of resistance already in Helicoverpa armigera. So I would expect, well, okay, let me think about it. In China, we have an example where we had early signs of resistance, I think about 10 years ago. And the resistance has gradually increased over the past 10 years, but it's still not what I would call practical resistance, not a severe problem. So we may have another 5 to 10 years, or maybe even more, if we're lucky, before it's a serious problem. Now, the early signs are already there, so it's a question of degree.
Thank you. There are actually some very interesting questions related to F2 hybrids. This one is from Alabaksh, and there's a question from Katagiri, Dr. Katagiri, and there's also a question from Dr. Raja. Now, these questions are from Alabaksh and Dr. Katagiri are asking almost the same question. It's on the yield compromise of growing F2 hybrids in cotton. The question of Dr. Raja is F2 seeds, is it an option for pink bollworm management in India?
So for the future, possibly. But as I said, the problem in India is that the refuge strategy is no longer likely to be effective with the currently available BT toxins, because the resistance is already very common. The resistance is high now. The refuge strategy, which can be implemented with the F2 hybrids, is kaput until we have toxins against which the pink bollworm is not resistant. So for pink bollworm, it's too late for F2 hybrids to help in terms of the resistance to Cry1AC or Cry2AB.
Thank you. Dr. Tasawwur Malik from Pakistan is asking how to handle resistant populations of pink bollworms, and Dr. Rajesh Patil from India is asking a similar question. What is the future of pink bollworm resistance management in India?
I think for pink bollworm, as I mentioned before, we need to find some novel toxins for new kinds of BT cotton. Otherwise, I would say we're out of the resistance management business for BT cotton, because we don't have BT cotton that works very well. Unless you were talking about an area of India where the resistance was not common yet, I think there still might be some, although they're pretty limited. And in those places, if the resistance isn't common, then the same strategies used elsewhere could work.
Thank you. There is this question from Dr. Tabib from Bangladesh. He's asking, is there any effect of natural enemies of cotton insect pests and resurgence of new pests due to cultivation of BT cotton in the ecosystem?
That's a great question. So I can speak to the situation in Arizona when insecticide use was reduced, critically against pink bollworm, but this also helped to reduce use against other pests. Then that increases the populations of natural enemies. So basically you could run the pesticide treadmill in reverse. So the less you use the insecticides, the less you kill the natural enemies, the less insecticide you need. So that's been a great benefit of BT cotton in Arizona.
Thank you. Dr. Y.G. Prasad, who is the director of the Central Institute for Cotton Research at Nagpur in India, is asking what line of action is best in tackling pink bollworm in India, at least in the short run. And he's also asking a second question, is mass trapping an effective strategy at flowering and in the late season? How does it compare with mating disruption?
Okay, I'll answer the first one, although I think I said before, I think now we're back to integrated pest management, maybe with a vengeance, with more zeal, with more belief from farmers because they know it's absolutely necessary now. And the second question was, say it again.
He's asking, is mass trapping an effective strategy at flowering and in the late season? And he's also asking, how does it compare with mating disruption?
Okay, great question. I don't know how effective mass trapping is. I imagine it can help a little bit. I'm pretty well convinced that the sterile releases, at least in Arizona, had a positive effect. I've also seen lots of field data that mating disruption with the female sex pheromone is also effective. So I think the mating disruption with female sex pheromone is a great idea to include as part of integrated pest management.
Thank you. Dr. David Stelly is asking, so according to the observed effects, I infer that a very good strategy for India would be to knock down resistance levels using a sterile release program, and once reduced to sufficient level, to implement a refuge-based strategy in addition, not before reducing the BT resistance level. Can you comment on what an optimal timeline and BT resistance threshold might look like?
I think it's too late to knock down the resistance. And I should say, I was impressed by the ability to release 11 billion sterile moths in Arizona. We have about 100,000 hectares of cotton. So in India, let's see if I can do the math. I think you have 100 times, the hectarage is about 100 times greater. So we would need a trillion, or like a trillion sterile moths. That's why logistically it's not reasonable overall for the nation. Certainly not without a new kind of BT cotton that would knock down the population. Even in Arizona, with a limited area of cotton that we have compared to India, it was very important to have BT cotton that was highly effective and that was used for a decade. That brought the population size down by about a factor of 100 before the eradication program was attempted. And once the population size was much lower, then it was reasonable to try to outnumber the wild population with steriles.
Thank you. This question from Peter Tan is actually very interesting. He's asking, how do you relate the soil health presentation of Dr. Lal to the presence of insect pests such as the pink bollworm? What implications would his presentation have on your work?
That's great. I'm so glad he asked that, because I was thinking about it during Professor Lal's presentation. Then I get wound up in my own world, but I think there is a definite connection there. So when we use BT crops or other transgenic crops, if we can figure out a way to use them sustainably, and for example, using them in combination with steriles and with refuges, or any other way we can use them sustainably, then we can reduce those other inputs, the insecticide in particular, and that helps to produce a sustainable system, and that's going to promote soil health. So again, we could get on a positive feedback system where we deplete the soil less and we need less inputs. So there's a real connection there. I think he had a great message.
Thank you. There are actually two questions that are related. One is from Dr. Rauf Khan from Pakistan, and the other from Dr. Amuda from India. They're asking, are there any other toxins being assessed for resistance to the pink bollworm if it develops resistance to the existing toxins?
Absolutely, yes. We have published results with what we call MOD, or modified BT toxins, that were genetically engineered by my collaborators in Mexico, Mario Soberon and Alejandra Bravo, and they do kill pink bollworm that are resistant to Cry1A and Cry2 toxins. So in the lab, that works great. there aren't any Bt cotton plants yet that produce those. There are other Bt toxins, naturally occurring Bt toxins, not genetically modified, that also kill pink bollworm resistant to Cry1 and Cry2 that's been used already. So the toxins are there. What we need, I think, is the will and entrepreneurship to get those in cotton plants that would be available to growers. And I would love to see that. And if someone knows a way we can promote that, I'm all in.
Thank you. Dr. Sabesh is asking two questions, but I'll read the first one first. His question is related to the land holding size in China, India, and the United States. He says in China and India, we have small land holdings, and in the United States, the land holdings are large. How does it impact resistance?
It's a great question. So the small land holdings, keeping everything else the same, are better for managing resistance when you're using the refuge strategy, because the refuge strategy requires the moths to move between Bt and non-Bt cotton. So if you have very large fields and the moths are not readily moving between Bt and non-Bt cotton, the refuge strategy is not expected to work as well. So those smaller holdings can be very beneficial for delaying resistance. And I think that plays a role with Helicobacter armigera being slower to evolve resistance. With those small holdings, you have the moths moving back and forth between Bt cotton and non-Bt varieties of other crops. So generally, the smaller holdings are actually better for managing resistance.
Thank you. Dr. Sabesh is asking another question. It's about refuge air. He says farmers lose some produce when they practice refuge air. How can this be offset?
That's a great question. Sometimes they lose less than you might think. So the best example of that is not with cotton, it's with Bt corn. It's very well documented in the United States that the European corn borer populations have been reduced regionally. And so it turns out that the economic benefits are actually greater in the refuges. Now, this is when the pest has been suppressed regionally by the Bt crop. When the Bt crop works well, that's what happens. The population density in the whole area declines, and that general decline means that the non-Bt refuge won't be damaged as much. So that's also going on with Bt cotton in China, at least in terms of pink bollworm damage. So that means you lose less on the refuge, and if those non-Bt seeds are less expensive, that's where the economic benefit can come in for growing the refuges.
Thank you. Dr. Mohan from India is asking, have you tried synergism between Bt cotton plus integrated pest management measures to break the pest cycle similar to what you have described as Bt plus sterile moths?
I'm not following.
He's trying to compare both the methods, which is Bt plus sterile moth technique with reference to Bt plus integrated pest management. He's asking, how do they compare?
Okay. I guess using Bt with sterols, that could be considered integrated pest management. The goal in the program that happened in the United States was eradication, which really isn't sort of by definition integrated pest management. But you could use sterols as just one more technique as part of integrated pest management, not necessarily to eradicate, but just to lower the population below economic threshold, which would be more in line with the IPM philosophy.
Thank you. Mr. Rajiv Barwa from India is asking this question. He says India uses hybrid cotton, which are hemizygous, whereas rest of the world uses homozygous Bt cotton. If India would have adopted the homozygous, like the homozygous varieties, would the situation have been different with reference to resistance in the pink bollworm?
It's a great question. I can guess, but I'm not sure. I think the key difference in the outcomes that I see is the presence of the refuges. So I would guess that no, it wouldn't have been better if India had used the homozygous rather than the hybrids. That's my guess.
There are actually many questions. Can I request you, Professor Tabashnik, if you can take a few more? Maybe it might take another 5-10 minutes.
Yes.
Thank you so much. This one is from Dr. Michel Folk. He's asking, to the best of my knowledge, cotton acreage has substantially decreased in Yangtze River of China since 2010 for lack of profitability. Could this trend be an additional explaining factor of the reduction of resistance phenomena?
Okay, that's a really good point. Well, okay, so the pink bollworm feeds primarily on cotton there. So that reduction in cotton probably isn't a primary factor reducing the resistance of pink bollworm. However, that could be very important in delaying resistance in Helicoverpa armidra, because that pest is using all those other crops. And as cotton becomes a smaller and smaller part of its diet, then that slows the evolution of resistance, because in effect you have a high percentage of refuges. So thanks for mentioning that. I think it's a great point.
Thank you. Dr. Ed Barnes is asking, as BT use in other crops increases, will the potential for resistance rates to increase as well? Any way to combat that if this is true?
I think, yes, it's true. When you have a pest that's feeding on several crops, all of which or many of which are producing BT, then you're reducing the refuge percentage and you can expect acceleration of resistance. We have an example in the United States, Helicoverpa zea, a close relative of Helicoverpa armidra. It feeds on BT cotton and feeds on cotton and corn, and both a high percentage of both crops have BT in the United States. It's evolved resistance widely in the United States to Cry1 and Cry2, and now VIP3 is the only BT toxin used in commercial BT crops that's effective against that pest in wide areas of the United States. So absolutely, yes, that's a problem. And we see the same problem in Brazil, where it's even worse because there's 3 crops, cotton, corn, and soy, all of which have BT varieties being popular.
Thank you. Dr. Negam is asking this question. What is the future of GMO cotton? He's from Egypt.
I think the future of GMO cotton is bright. I think of GM cotton as just GM in general, just being a tool, a technique, a way of improving the crop. And I should say, I don't see it as a panacea. I don't think any one approach is a panacea. It's just adding more tools in our kit. And just like any tool, it's about the tool itself isn't good or bad, it's how you use it. So our challenge is to find wise, environmentally friendly, safe, healthy, and sustainable ways to use GMO cotton and other GM crops.
Thank you. Dr. Shrikant Patil is asking, can the promotion of organic cotton and also like a limited mandatory spray of pesticide in conventional cotton, can it improve the situation to manage resistance to in the pink bollworm?
Yes, yeah, because the organic cotton is not going to be BT cotton, so that increases the percentage of the refuge, so that helps to manage resistance. So absolutely, yes.
Dr. Satnam Singh from India is wondering why Helicoverpa armigera and spotted bollworm are taking time for resistance development. And he's also wondering, is it because of the recessive nature of the alleles that resistance is taking a longer time in these pests?
So Helicobacter armigera, I know pretty well, and we've talked about that. It's eating many different hosts, and most of those don't have BT, so that's loaded. Spiny bollworm, you'll have to tell me. I don't know much about it. I'll just say though, in armigera, surprisingly, the resistance is not recessive. Not recessive. So even though it's not recessive, there's such a large proportion of non-BT hosts, such a large refuge. That's what's slowing it down. So what about, I don't know, spiny bollworm. What does it eat? Plants other than cotton?
It actually eats ladyfinger, most malvaceous crops. It does have a reasonably good host range, but not very parallel with cotton, not something that grows in parallel with cotton. But it does have.
Okay, well, sorry, I think that's a good topic for further investigation. I don't know if is it being monitored closely for resistance? Would it be noticed? Those are questions for a good follow-up.
This question is from Dr. Sen from India. He's wondering if pink bollworm eradication program is safe for the ecosystem, especially with the millions of releases of moths.
Yeah, I think it's very safe. Certainly much safer than the massive use of insecticides that was going on before. Those billions of moths are not harmful to the environment that were released. It's actually done now. No more sterols are being released now, but a very safe technique, very environmentally friendly, certainly compared with what was going on before, which was heavy use of broad spectrum insecticides.
Thank you. Mr. Rajiv Barwa is asking, he's from India, he's asking if the innumerable number of BT cotton hybrids of long duration, have they impacted resistance? How do they impact resistance?
So hybrids that are grown over a longer season?
Yes.
Generally, the longer season you have, the more you're encouraging the pink bollworm, because throughout the season it builds up. So if you have a long season hybrid, then you're encouraging it. And I think that's been one of the important components of the IPM, is to avoid those long season hybrids, go with shorter season. It gives pink bollworm less time to build up. And I forgot, it's something that I want to mention that was used in Arizona along with the techniques I've talked about before, which is destruction of crop residues to reduce overwintering of pink bollworm in cotton fields. That's an important IPM technique. It was part of the eradication program. I think farmers in India are using that now, and that can be very effective.
Thank you. This question is, can Bolgar-3 with Cry1AC, Cry2AB, and WIP3A, can it be a part of solution to control the resistant pink bollworms in India?
I'll tell you that I have data on WIP3 against pink bollworm from the lab. It's not very effective. So that's the problem. Otherwise, it would be great. Just switch to the Bolgar-3. I have not tested the WIP3 containing plants. I would like to do that, but based on the LC50, the amount of WIP3 that it takes to kill pink bollworm when they're tested in bioassays, WIP3 is not very effective, so I don't think it's promising for controlling resistant pink bollworm in India.
Thank you. Dr. Shrikant Patil is asking if there's a fitness cost associated with resistance in the pink bollworm, and how does it have an impact on the survival of these larvae on non-GM crops, including organic cotton?
That's a great question. I'm really glad he's asking that because the refuges, if there's no fitness cost, all they can do is slow down the evolution of resistance. But if there's a fitness cost, then in refuges, you can actually select against resistance. And so that fitness, pink bollworm, virtually all the experiments we've done, it does have the resistance to BT cotton does have a fitness cost. So in China, it was not just increasing the refuge percentage, but that interacting with the fitness costs that allowed for susceptibility to be restored. I also want to say, remember that the percentage of individuals in a population in China that was resistant, the average was only about 2.5% when the reversal occurred. So that's a low percentage. Once you get much higher, say over 50%, it's much more difficult for the resistance to be reversed, even if there's a fitness cost. That's because compared to the selection for resistance that's caused by Bt cotton, the selection against resistance caused by a fitness cost tends to be pretty weak. So you can select for resistance really quickly, but going down, usually not so fast because the fitness cost tends to be relatively small.
Thank you. We just have a couple of questions before we end this session. So thank you for your patience. Dr. Rishi Kumar is asking two questions. One is, what is the role of refuges after resistance has already developed?
What is, say it again,
what is the role of planting refuge after resistance has already developed?
After resistance has developed, they're really not useful, not in an immediate sense. Now, over a long period of time, let's say maybe 10, 20 years, if people stop planting the Bt cotton altogether, susceptibility might be restored over a long period of time. But after resistance evolves, refuges are not particularly useful.
Thank you. And his next question is, how does diapause in the pink bollworm affect resistance development?
That's a good question. Well, in temperate regions, the overwintering is kind of a bottleneck for pink bollworm. So that's a good time to attack it. And so for example, destroying the crop residues and anything else you can do to hit the emerging populations in the beginning of the season after diapause, because the populations are relatively small then. So mating disruption can be especially effective then, and also sterols can be highly effective.
And this is the last question from Dr. G. Balasubramani from India. He's wondering, why does pink bollworm infect only 1 or 2 loculi within the boll? Is it because the Indian Bt hybrids, are likely to have non-Bt seeds in them?
That's a good question. We'd have to, do the experiment to really know. I think, I don't know if the number of locules affected would differ depending on whether the plants are hybrid or not, but it's a really good question.
And just one final one, which I got just now. This is from Dr. Tasdeequr Rahman. He's asking a general question, which is jacids, which are leaf hoppers, is now a major pest in Bangladesh. Are there any Bt cotton varieties to control jacids?
That's a great question too. I know there are these there's a new kind of Bt cotton that controls some pests other than Lepidoptera and Coleoptera. I don't know if it controls jacids, but it would be a great thing to find out. This new kind is in the pipeline, hopefully be commercialized in the next 5 years or less. But I don't know if it controls jacids.
Thank you very much, Professor Tabashnik, for the brilliant presentation and for your extraordinary patience with so many questions. I can see there are some questions that are still coming in, but I don't think I take them. I've always enjoyed reading your papers, and like many of my other colleagues, we look to you for our inspiration. You are the leader in the field, and for us, you are a living legend. Thank you so much for accepting to speak and for the brilliant, for this brilliant exposition. I'd now like to request Mr. Kai Hughes, the Executive Director of the ICAC, for his final word. Kai.
Well, thank you, Bruce, Dr. Tabashnik, for it was a really brilliant presentation, and I'm grateful for you giving that. As a non-scientist myself, working in cotton and being surrounded by people like Dr. Kranthi and Dr. Sanya Kranthi, I try to make a daily advancement in cotton research knowledge. And I think that both you and Professor Lal have used up a couple of years worth today. So that's, so I'm deeply grateful for you for that, because that means at least I can have a few days off over Christmas and relax. But from all of us here at ICRA and the ICAC, everyone who's attended today, I'd like to thank you once again. And we look forward to meeting you and talking to you again at, hopefully, at the World Cotton Research Conference in Egypt next year. Thank you.
Thank you very much. Thank you, Professor.